So my name is Jonathan Chou. I'm one of the medical oncologists here, and like Dave, I see patients and also run a lab. And this topic on targeted therapies is one that's very near and dear to my heart, both because I think it highlights the tremendous progress that we've made in thinking about how to integrate genomics and what we know about the mutations and the building blocks within prostate cancer, but also because it highlights the tremendous amount of work that still lies ahead for us. So of course, when I was putting together this talk, I asked ChatGPT, what is targeted therapy for prostate cancer? And I thought ChatGPT actually gave an excellent response, much better and much more eloquent than I could have given. And what ChatGPT said was, in oncology, targeted therapy refers to cancer treatments that are designed to specifically identify and attack molecular features of the cells. We'll talk a little bit about what those molecular features are, rather than broadly killing all rapidly dividing cells like traditional chemotherapy does, okay? And so the key idea here is that we're going to need to understand the unique genetic mutations or abnormal proteins that might drive the prostate cancer to grow, and that we're gonna build therapies that are targeted to interfere with those specific abnormalities. And so how does this actually work? So at a very high level, what we're thinking about are what are the mutated genes in prostate cancer, okay? And I've just listed a couple. There's a gene called PTEN that's commonly mutated. The androgen receptor, which is what testosterone binds to, can be mutated in prostate cancer. There are gonna be overexpressed receptors. So we just talked, Ivan just talked about PSMA targeting in prostate cancer, so proteins that are outside on the surface of the prostate cancer cells that we might be able to target, and then various different signaling pathways that might control growth, and we'll talk a little bit about what those might be. So I think really to understand what targeted therapy is, we have to go back to the basics of biology. And so just to remind everyone again that our cells, including the cancer cells that are in patients' bodies, are made up of DNA, RNA, and proteins, just like every other cell that's in your body. These are the A, T, Gs, and Cs that form this double helix. And essentially what ends up happening is that mutations in the DNA, in the cancer cells, either lead to uncontrolled proliferation and also lead to the decrease of what's called apoptosis, which is normal programmed cell death. So you have too many cells that are growing and not enough cells that are dying. And we think that this really stems from, at least at the most basic level, to mutations that are happening in the DNA that you accumulate during the course of your lifespan. So remember, every cell in your body needs to divide over the course of your lifespan. There are mutations can happen randomly, and some of the mutations, unfortunately, either lead to one of these or both of these processes. And so these mutations in the DNA ultimately affect the RNA that's being transcribed from the DNA, and then ultimately make abnormal forms of proteins that can then affect the growth processes. And so we really need to understand all levels of this biology, the DNA mutations, the RNA transcription, and then also this translational machinery, really to understand and identify new ways of targeting prostate cancer. And really, you know, most of the chemotherapies that we've known for like 50, 60, 70 years, they primarily target the ability of the cells to replicate. And that's why many of the side effects of chemotherapies are related to rapidly dividing cells in our bodies. So bone marrow suppression causing anemia, low platelets, low white blood cell counts, because those cells are being constantly replenished in the body. Hair loss, because hair is constantly growing, and also GI symptoms. So, and it gets even more complicated than what I just showed you. Obviously, this is a cartoon, and I have to talk about this in 10 minutes. But these strands of DNA are actually wrapped around proteins called histones, which are organized. They're then curled up and wrapped all around in these things called chromosomes. And so at every single level of the molecular biology, there's something really interesting that goes awry in the prostate cancer cells. So in order to identify those targeted therapies, we've had to engage in a huge effort. And this is really highlighted, UCSF was one of the main leaders and drivers of this effort that was sponsored by the Prostate Cancer Foundation and Stand Up to Cancer to collect biopsies from over 400 men who generously donated tissue for this study. And we had to understand the landscape. What are those mutations in the DNA, RNA, and protein levels? And this was led by Drs. Eric Small and Felix Fang to perform some of the first whole genome sequencing and RNA sequencing and whole genome bisulfite sequencing to identify all those changes on patients who had metastatic and advanced prostate cancer. And this has led to some of the most seminal publications in our field that have really highlighted in work that Dr. Quigley, Dr. Agrawal, Dr. Chen, who you've heard earlier, Dr. Zhao, really to identify what are the things that we need to be studying and we need to be targeting. I'm going to give you an example, one that's now been recognized over 10 years ago, in that these are looking at men who had prostate cancers, metastatic prostate cancer, and the frequency of mutations that were being passed from either mom or dad to the patient. And so what was really surprising here was that in this study, about 11 to 12% of men actually had a heritable genetic mutation that was passed from one of their parents. And when we looked at what those heritable mutations were, almost half of them were in a gene called BRCA2. And for those of you who know what BRCA2 stands for, it actually stands for breast cancer susceptibility gene. And so this is a gene that originally was identified because it was being passed from mothers or fathers to their daughters. And it took actually another 20 years for us to actually realize that this gene also increased men's risk for prostate cancer. This table over here shows you a list of different mutations that cannot be necessarily passed from parent to child, but mutations that accumulate only in the prostate cancer cells themselves. And this is actually taken from Dr. Quigley, who's on faculty here now, from one of the seminal papers that our group had published. And I'm not going to talk about every single one of these gene mutations. These are all different genes, but every single row is a different patient as part of the cohort. And I'm showing you about 40 of those patients out of the 100. What I want you to appreciate from this graph is that there are some genes, like P53, where there's quite a few patients who have this particular mutation. But then there's a lot of genes where, like MSH2, which is one of the genes that Dr. O just talked about, it happens not in every single patient, but in a patient who has that MSH2 mutation or that MSH6 mutation, those are patients who are responsive to the anti-PD-1 immunotherapy that we just heard about, right? And so what I think we can appreciate as we go down every single row here is that every patient is pretty different, right? You don't see the exact same pattern in every single patient. And I think what that really tells us is that the building blocks for each individual patient's prostate cancer are just so different. And so instead of trying to treat everyone the same, I think what we really need to do is understand what do these mutations do and what are the Achilles heel susceptibilities that having a mutation in MSH2, for example, what's the susceptibility? Well, it generates a lot of frame shift mutations that can then be neoantigens that the immune system can recognize. So that understanding actually tells us that these are patients who might be really responsive to that checkpoint immune therapy. If we've done the trials, if we take all comers, right, people who don't have this mutation and we put them on, the response rate is like 3%, right? So we don't need to waste 97% of people's time, but for the 3% that have this mutation, the response rates to that therapy are tremendous. So let me give you another example with the BRCA mutation. We heard a little bit from Dr. Desai's talk about PARP. So patients who have a BRCA mutation, either BRCA1 or BRCA2, whether it was inherited from their mom or dad or whether it spontaneously generated in the tumor, benefit from PARP inhibitor therapy. So those are drugs like olaparib, rucaparib, and talazoparib that are now FDA approved for these particular mutant patients, right? And so What's remarkable, right, is that if you don't have one of these mutations, you are not going to respond. The tumor is not going to respond. So you're going to get all the side effects of the therapy and none of the benefit. And we've now seen that also for CDK12, like the right CDK12 mutation in blue, these biallelic mutations, more than half of these patients will respond to a PARP inhibitor. So we really need to be very thoughtful about the mutation in the tumor and how we actually target it. The final thing that I'll talk about is that the genetics, those mutations in the cancer cells, can actually alter the collection of proteins that are on the cell surface. And this is something that our lab has been really interested in thinking about. How do we identify those proteins on the cell surface? Because those are the proteins, like PSMA, that if we can figure out a protein that is unique to the prostate cancer cell, we can actually design both a diagnostic, and we heard a little bit about PSMA PET scans, but there's a lot of different other proteins that we can think about for our diagnostics, and then also drugs like PSMA lutetium. And so here's an example of a patient that I had where the PSMA is negative. It would have showed up in this gold color over here. You see a couple of these pink highlighted T cells here, but really this is a PSMA negative patient. It's also negative in STEAP1, that's the xaluritamig or AMG 509 that Dr. O just talked about. But take a look, this is a patient who has very, very high expression of DLL3, and Dr. Agarwal is actually leading one of the several DLL3 targeted trials here at UCSF for people who have a tumor that is DLL3 expressing, but perhaps not PSMA expressing. And so we now in our oncology toolbox, it's such an exciting time to do and treat cancer, because there are so many different tools now at our disposal. And we heard a little bit about radioligand therapies, we've heard about T-cell engagers, and then trials looking at CAR Ts and T-cell receptors and also antibody drug conjugates, all within the trial portfolio here at UCSF. And so here is really the holy grail, the goal that we have, right, is using our understanding of the genetics and genomics within each individual patient's tumor to really guide how we order our therapies, which therapies are appropriate and which therapies aren't, so that we can really actually match, right? If you have a tumor with a mismatch repair mutation, you get the blue therapy, which is checkpoint therapy. If you have a tumor that has a BRCA mutation, you get the pink therapy, et cetera, et cetera. So there's so much more work to do because, you know, obviously when I showed you that table, there's lots of different flavors of prostate cancer. And so this is what the kind of the next generation, the next wave of treatments for prostate cancer are really gonna look like. It takes a huge team and dedication and a lot of funding and efforts to do this. Highlighting a couple of people, it takes cross-disciplinary efforts from urologists to radiation oncologists to radiologists, nuclear medicine docs, medical oncologists. You've heard from all of them here today. We have such a great team here at UCSF to be able to really advance prostate cancer treatments for the next generation. So with that, thank you very much for your attention.